Welcome everyone. Here is another question from GATE 2020 Biotech PYQ series. The question is, protein A and B form a covalent complex. Gel filtration chromatography of this complex showed a peak corresponding to 200 kilodalton. SDS-PAGE analysis of this complex with and without beta-mercaptanol showed a single band corresponding to molecular weight of 50 and 25 kilodalton respectively. Given that the molecular weight of protein A is 25 kilodalton, the molecular weight of protein B is, we need to find out the number in kilodalton. So let's try to understand what all is given here. So there are three techniques which they have talked about, and they are trying to understand the complex formation by the protein A and protein B. So the first method which they have used is gel filtration. This method separates the biomolecule on the basis of molecular weight in native condition. Now, with the help of gel filtration chromatography, a peak corresponding to 200 kilodalton was observed. That means the protein A and protein B is forming a complex. And as mentioned, the complex is covalently bound. Now, what are the different ways the two proteins can covalently bond? One of the most prominent ways in which the two components can bind is with the help of the disulfide bond. However, there could be other ways, and we'll get to know whether those are bound with a disulfide bond by making use of beta-mercaptanol, which is mentioned in the other part of the question. So gel filtration is showing 200 kilodalton. Now let's see the other part of the question. It says with the help of the SDS-PAGE, it's a method where we separate the biomolecule on the basis of molecular weight. Under denaturing conditions, we make use of a sodium dodecyl sulfate, which is a detergent, which denatures the protein by removing all sorts of non-covalent interactions. And the use of beta-mercaptanol or dithiothriotol is to break the disulfide bonds. So in the presence of the beta-mercaptanol, a band of 50 kilodalton was observed, while in the absence of beta-mercaptanol, a band of 25 kilodalton was observed. The other alternative for beta-mercaptanol is dithiothriotol. So as you can see, SDS-PAGE analysis with beta-mercaptanol showed a single band corresponding to 50 kilodalton. Now, what does that tell you? There are two proteins, A and B, and those are not held together with the help of disulfide bond. Why I'm saying so? Because if I'm using a beta-mercaptanol, and if they are held together with the help of disulfide bond, then the beta-mercaptanol will cleave those two bonds, and you will see bands corresponding to A and B, because the other part of the information says the molecular weight of protein A is 25 kilodalton. So it's given A is 25 kilodalton, and if these two would have bound with the help of the disulfide bond in the presence of the beta-mercaptanol, that have resulted in the cleavage of that disulfide bond, and we should have got a single band of 25 kilodalton. But what we are getting here is a 50 kilodalton band on SDS-PAGE analysis in the presence of the beta-mercaptanol. That means these are joined together covalently as mentioned, but not with the help of disulfide bond. So let's check what is happening in case of SDS-PAGE in the absence of beta-mercaptanol. These two processes are almost same. The only difference here is absence of the beta-mercaptanol. And we all know the function of the beta-mercaptanol is to cleave the disulfide bond. The use of the beta-mercaptanol tells you whether the complex formation or whether there is a formation of disulfide bond in the protein. Now, in the presence of the SDS-PAGE, where no beta-mercaptanol is used, a single band of 25 kilodalton is observed, which is quite surprising. So if I'm not making use of beta-mercaptanol, the complex should have intact, and we should have got a band corresponding to 50 kilodalton. So the information seems wrong here. Now let's solve the same question. If it would have given that SDS-PAGE analysis with beta-mercaptanol is giving 25 kilodalton, while without beta-mercaptanol, it is giving 50 kilodalton. This time I'm changing the molecular weight in the presence of beta mercaptophenol and in the absence of beta mercaptophenol. Now, this makes much more sense. The reason being, if I'm getting with the help of size exclusion 200 kilodalton and with the beta mercaptophenol, I'm getting a single band of 25 kilodalton, and the information is already there that the molecular weight of A is 25 kilodalton. That means protein A is covalently bound with the protein B with the help of disulfide bond. So what will happen if I'm making use of a beta mercaptophenol? This bond will be cleaved and we'll get a single band. We'll not be able to distinguish protein A and B on the SDS page. The reason being both of them are having a same molecular weight. Now, what is happening when I'm not using beta mercaptophenol? I'm getting a band of 50 kilodalton, which makes much more sense because what will happen when I'm not making use of a beta mercaptophenol, disulfide bond will not be cleaved and I'll get a band of a AB complex, which is 50 kilodalton. So in this case, this will be cleaved, while in this case, the disulfide bond will remain intact. Now let's summarize the things and have a look how this protein complex is assembled. So you have protein A bound with protein B with the help of a disulfide bond. This is 25 kilodalton. This is also 25 kilodalton. However, the overall molecular weight of the protein is 200 kilodalton because just now I said in case of the size exclusion or gel filtration, we get to know the molecular weight of the protein in a native conformation, while in case of the SDS page, we get to know the molecular weight in a denatured condition. So if this is 50 and we all know the overall molecular weight of the protein is 200 kilodalton, what does that tell you? That there are additional 4 copies of this heterodimer. So you have AB, which is covalently bound, and then you have 4 units of this complex, and one subunit is associated with the other subunit with the help of non-covalent interaction. And this seems to be correct because in that case, when I'm making use of a beta mercaptophenol and running the SDS page, all these disulfide bonds will cleave and I'll get a 25 kilodalton band of A and a 25 kilodalton of B. Rather, 4 bands of A and 4 bands of B, which is 25 into 4 and 25 into 4. The overall molecular weight is going to be 200 kilodalton. Now, in the absence of beta mercaptophenol, what will happen? Of course, these non-covalent interactions will break. However, the covalent disulfide bond will not be cleaved because there is no beta mercaptophenol. And I'll get a band corresponding to 50 kilodalton, and those are going to be 4 copies. So 50 into 4, 200. And in that case, the justification seems to be correct. So overall, the initial part of the question was not correct. And considering this is the question, we can mention the molecular weight of the protein in this case is also going to be 25 kilodalton. I hope this question and answer is clear to each and every one of you. Thanks for watching the video.